insights, and we're so happy that you're going to be taking time for questions. We saved the best for last. So to all of our participants, if you have questions for any of the presenters, please put them in the Q&A pod, and with that, we'll go ahead and get started. Our first question is for Dr. Tyner. Is there a preference in configuration of the tubes? For example, the distal tip being closed-ended or open-ended? Delivery of the entire dosage seems to be easier with open ends.
Thank you for that question. We request that companies perform risk assessment of the formulations and design in vitro testing accordingly. This should include justification of the selection of the tubes used in the test. As the question mentions, open ends can allow for freer flow of the material and could help in the design of a worst case scenario. However, in some cases, as Mamta mentioned, there is a limitation of the configurations available, which FDA recognizes. Please make sure that the study design, the rationale, and the justification is included in this.
Great, thank you. Another question for you. For NG recovery testing, should we do N12 for each type of the NG tube? For example, PVC, silicone, or PE. Should we select one based on the type of risk?
So for the BE assessment, it is performed with 12 units with one tube configuration. The quality assessment is performed on all three of those configurations, and we also would recommend N equals 12. Please do note that the three types of tubes do not have to be based on polymer material, as the question suggests, but can be varied by eyelets, closed open distal end, and what have you. Also know that it is the company's burden for the study design and justification. For example, if there are repeat administrations, it might be acceptable to use just 4 tubes and have 3 sequential administrations in one of those tubes to combine both the repeat dosing and the recovery studies and get N equals 12.
Great, thank you. And another question for you from the audience. Are there any recommendations for tubing material? Tubes usually contain plasticizers, and they're not sure if the tube should be free of hazardous.
Right, another fantastic question. So we do not require specific tubing materials. In some cases, the reason for that is that these different materials are not available for different tube sizes or what have you. So we do request 3 different designs, and that can include tubing material. So if you're able to get 3 different materials that have the different plasticizers, that's fine. But you can also vary based on ports, number of eyelets, etc.
Great, thank you, Katherine. So, reminder to our audience to go ahead and enter your questions into the Q&A pod. Our next question is for Dr. Kapoor. Is the syringe selection important when flushing the feeding tubes as much as it is important when the dose is given?
Thank you for the question, Lisa. So syringe selection may not be as critical if the package insert or the product-specific guidance does not mention a particular syringe. In some cases, it mentions the syringe. For example, for one of the products in my talk, there was a recommendation for use a catheter-type syringe. Otherwise, you can use any syringe that is suitable for use with tubing materials. Thank you.
Great, thank you. We have another question for you. An NG tube study is to be done with 3 different pHs. Can you suggest buffer or an acid base to adjust the pH of?
Thank you, Lisa. So for pH study with tubings, we do not recommend the use of buffers just because we want you to mimic the clinical conditions in the clinical setting. So we do suggest that you do the pH adjustment using acid or base. That's how we want you to do it instead of using any buffers. Thank you.
Okay, great. We have another question for you. As per the PSG recovery testing, PSD, acid resistance and sedimentation volume, is to be done for all 3 pH in all 3 tubes. can we use the worst case scenario as polyurethane to perform all the tests and perform recovery testing as well as performing all the pH and all the
Yeah, it seems like a lot of testing, but if you see the PSG for this, any product, you will see that acid resistance you don't have to do with the 3 kind of tubing material, and nor does recovery testing for the BE perspective. It's only for quality perspective that we recommend 3 type of tubing material. So if you look into the guidances more closely and the PSGs, you will get more clarity on this. However, with this question, we cannot use polyurethane as a worst case because polyurethane has the widest lumen. On the contrary, silicon has the narrowest lumen because of the thickness of its wall. That can be still considered as a worst case with respect to tubing material. Overall, I would say that your choice of worst case should be justified when you submit your application to the agency, because it's very much formulation dependent and it depends what the guidance or PSG recommends for that product.
Thank you, Dr. Kapoor. Our next question will be for Commander Cui. So the first question is, when using systemic PK data to draw conclusions about local availability in the GI tract, what PK metrics are most valuable for the bioequivalence evaluation?
Thank you for the question. The most valuable PK metric for evaluating local bioavailability of drug is partial AUC. Because the mesalamine is locally, for example, mesalamine is a locally acting drug, so the primary target site is colon. However, the absorption site is along all the GI tract. So the to ensure that demonstrate bioequivalence at a local action, we have to pick up the PK AUC metric that is relevant to the local absorption for the action side, which is that's why the partial AUC, for example, for mesalamine delayed release product, the partial AUC 84 to 2048 was picked is because that time interval is related to absorption at a colon. Thank you.
Thank you, Commander. We have another question for you. What is the acceptance criteria for sucralfate pepsin inhibition?
That's a good question. We do not have a very specific criteria in the PSG. So that's treated as a review issue. So when you submit the data, we will compare the test and reference. So if your data showed a very close comparable inhibition on the pepsin activity, then that's considered acceptable.
Great. Our next question for you is, if plasma concentration can be measured, can we only conduct PK studies to ensure the bioequivalence for locally acting GI?
That recommendation, although it's product specific, however, normally we for local acting product, we do recommend both PK and in vitro dissolution. The reason is if it's a systemic acting drug, PK will be the most sensitive, reproducible measure. However, if it's a local acting, then PK concentration not necessarily to reflect the concentration at a local site. So in this case, in order to ensure the equivalent delivery to the action site, agency often recommend another method to, in combination with the PK, to support bioequivalence. In many cases, in vitro bioequivalence study. That's why if you look at mesalamine product, fitting this category is local acting, but PK can be measured. So we recommend both the PK study and the in vitro dissolution over the range of a pH representative of GI tract. With that, we think that bioequivalence at a local site can be ensured. Thank you.
Great, thank you. And another question for you. For rifaximin, why is the clinical endpoint study recommended in addition to in vitro dissolution and in vivo PK study?
That's a very good question because if the I just mentioned mesalamine. So this rifaximin is a local acting drug, but with a very low solubility and permeability. So because that, even you have a equivalent, you have a comparable dissolution profile and a similar PK, anything change if it's not Q1, Q2. Anything, even either type of excipients or amount of excipient changing the formulation could affect the metabolism, absorption, and even antibacterial activity at action site. Because of that, so even you have an in vitro study and have a PK, we still recommend clinical endpoint study because the drug substance is low solubility and low permeability. Thank you.
Thank you, Commander. And our next question is for Dr. Tyner. When performing an NG tube study, it is assumed that the delivery of the dose is performed by injecting with the syringe by the use of a plunger and not by gravity.
This is a great question. So correct, pressure may be applied to the syringe, and oftentimes you're not going to have the product be delivered unless you do that. Now, if you really have to push down on the plunger, that might be indicative that you have a clog or something else going on. And I'd also caveat that you should be following administration conditions that's found in the product labeling. So if the label did in fact say that you should not use the plunger but only let gravity do the administration, then that's what you would follow. I would refer you to the product-specific guidances, because if it did have something like that, we would be calling that out in the product-specific guidance.
Great, thank you. We have another question for you. Your talk only covers NG and G tubes. What about products that are delivered through other enteral feeding tubes, such as for Dr. Tyner, if you, I can go ahead and, I'll go ahead and reread the question for Dr. Tyner. Your talk only covers NG and G tubes. What about products that are delivered through other enteral feeding tubes, such as?
Great, thanks. That's another great question. So the reason why my talk covers mainly the NG and G tubes is because those are the ones that we most commonly see. However, if the label did say to administer through another type of tube, such as a J tube or an NJ tube, please follow that and use the same type of criteria and the same testing recommendations for those tubes.
Great, thank you. And we have a question for Dr. Kapoor. A product has a product-specific guidance, but there is no in vitro enteral feeding tube testing recommendation in the PSG or in the product package insert. How do I?
That's a great question, and that's what we want the firms to think about. If there is no testing in the PSG or the PI, please communicate with the agency through suitable channels such as control correspondence, where you can ask this question, and based on the product and the risk assessment, we can provide you with recommendations for the testing. Thank you.
Great, thank you. Another question for you. If the 6F silicone tube is not available in my country, how can I provide enteral feeding tube testing with 3 tubing?
I think the answer to this lies in, how much Katherine has focused in her talk about it's not just the material, but also the design. So if you cannot find silicone tubing 6 French, we suggest that you use different designs. If it's a nasogastric tube, for example, a tube with open or closed distal ends or with number of different eyes or ports. So it is not only the material that can make a difference in the clogging behavior. but it is also the design of the tube. So if possible, you can look into different designs. If you have access to the global market, you can go through that channel. But otherwise, we would suggest you to use whatever material is available. Try to use different designs to make it like 3 different designs for NG tube or G tube. Thank you.
Thank you. And we have a question for Commander Cui. For rifaximin, if the test product is not Q1 and Q2 same as the RLD, should in vivo PK and clinical endpoint studies be conducted on both?
Thank you for the question. That all depends on if formulation is proportional or not. If 200 milligram the strength is proportional to 550 milligram, then if the Q1, Q2 not same, well, you only need to conduct a PK study on 200 milligram and a 500 clinical endpoint study on 550 milligram. It's because PK is not linear, so it at higher concentration absorption is a little bit saturated. However, if the formulation is not proportional and the Q1, Q2 not same, then you do need to conduct a PK study on both strength and clinical endpoint study on both strengths. Thank you.
Great. Thank you so much, Commander Cui, and also to Drs. Tyner and Kapoor. And with that, we're wrapping up our question and answer section. And we appreciate your time, and we appreciate all the participants staying.